During development and tumor metastasis, cells prepare to migrate by undergoing an epithelial-to-mesenchymal transition that loosens their connections to neighbouring cells. However, as Roberto Mayor from University College London explains, many cell types, including neural crest cells, subsequently migrate en masse to their final destinations, indicating that their intercellular adhesions aren't disrupted completely.
We were interested in how the neural crests migrate collectively, and at the same time they undergo EMT, epithelial-to-mesenchymal transition, because these two seem to be two different aspects of cell behavior. In one case you have an individual cell migration, and in the other you have interaction between the cells.
Mayor and colleagues, led by Sei Kuriyama and Eric Teveno, looked at the potential role of lysophosphatidic acid, or LPA, signaling in regulating the migration of Xenopus neural crest cells.
First, we look at the expression of all the LPA receptors. There are 5 different LPA receptors, and we found that the LPA receptor 2 was the only one expressed in the neural crest.
Knocking down LPA receptor 2 with antisense morpholinos inhibited neural crest migration, and grafting experiments showed that the receptor was acquired in the neural crest cells themselves rather than in their surrounding tissues. Kuriyama et al. Then looked at the activity of the receptor during embryonic development.
We analyzed directly the phosphorylation of the receptor, and we found that there was an increase in phosphorylation just before the neural crest started to migrate. So we mutated the putative phosphorylation sites, and we found that the mutation of a specific phosphorylation site not only blocked phosphorylation as we expected, but also blocked the ability of this mutated form to rescue the neural crest migration in embryos in which the wild-type receptor was depleted. So the mutation blocked the activity of the molecule.
Surprisingly, however, knocking down LPA receptor 2 had no effect on neural crest cell motility or chemotaxis in vitro.
So that was fascinating because we didn't know why you had this very strong phenotype in vivo but not in vitro. So one difference is that in vitro the cells can migrate everywhere, while in vivo there are physical constraints. The neural crest need to migrate around the eye, in the branchial arches, where there are a lot of other cells. So we thought that maybe the difference between in vivo and in vitro was because in vivo there was this physical constraint, and when you block this LPA, for some reason the cell could not migrate into this physical constraint.
To test this idea, Kuriyama et al. Examined the cell's ability to migrate toward a chemoattractant through narrow microfluidic tunnels. Wild-type neural crest cells could rearrange themselves to squeeze through the tunnels and reach the chemoattractant, whereas cells lacking LPA receptor 2 congealed at the tunnel entrance.
So we thought that maybe in order for the cell to migrate into very narrow tunnels, they have to behave more like a fluid, so can adapt to this small tunnel and migrate into this physical constraint. So that is why we decided to analyze the fluidity of this group of cells by measuring the correlation between the velocity of a cell and the neighbor cells. And we found that in the control cells exchange positions with the neighbors, while in the cells depleted of the LPA receptor, that was inhibited. So the cells did not exchange positions, they maintained the same neighbors, and we call that a more solid-like behavior.
The increased solidity of LPA receptor 2 deficient cells could be due to enhanced intercellular adhesion, limiting the cell's ability to exchange positions with their neighbours. Indeed, in an in vitro dispersal assay, control cells quickly scattered while receptor deficient cells remained tightly clustered.
The levels of cell adhesion protein that is called N-cadherin was increased at the cell contact in the LPA receptor 2 depleted cells. So in the normal neural crest cells, N-cadherin is at the cell contact very transiently, But when we block the LPA receptor, the N-cadherin remains at the cell contact because the endocytosis was inhibited.
The researchers reason that if this increase in intercellular adhesion is the cause of the decreased fluidity and migratory capacity of neural crest cells lacking LPA receptor 2, then the phenotype should be rescued by decreasing N-cadherin function or expression, or by enhancing the adhesion protein's endocytosis.
All this treatment, inhibition of cell adhesion, inhibition of N-cadherin, or promotion of endocytosis, rescue the dispersion of the cells in vitro, also rescue the migration into the tunnels, it rescues this fluidity behavior that we described, and finally, it rescues the migration in vivo.
However, completely abolishing N-cadherin-based cell adhesion also inhibits neural crest migration in vivo.
If you have complete individual cells, the cells do not respond to external signals, because in order for the cells to respond to the external signals, they need to be polarized, and the polarization depends on cell-cell contact, which is N-cadherin dependent. So you require a minimal level of N-cadherin to allow cell-cell interaction, which explains this collective behavior.
Kuriyama et al. Therefore think that, when followed by collective cell migration, EMTs don't result in a fully mesenchymal phenotype with a complete loss of intercellular adhesion.
So what we're proposing here is that for collective cell migration, the EMT is the acquisition of this more fluid behavior that allows the cells to migrate into physical constraints. We show this is true for the neural crest, but we believe that this is more general than the neural crest, and it's known that, for example, during metastasis, many cancer cells migrate as a collective, and they undergo EMT as well. And they are in an environment which is full of physical constraints. So we believe that it's very likely that this acquisition of fluidity is also the result of EMT in cancer cells.
Mayor and colleagues now want to investigate this possibility, but in the meantime, you can learn more about how collective cell migration requires an LPA receptor 2 dependent increase in tissue fluidity in the paper by Kuriyama et al., published in the July 7, 2014 issue of the Journal of Cell Biology.
Ciao.